version of it. Jamie's not able to join us today, so it's Don and I for faculty, and then the rest of you guys. And so I guess, Liana, we'll just go ahead and turn it over to you. You can introduce yourself a little bit. And if you guys have questions, go ahead and ask as we go through it. What we're going to do today is have Liana give her talk for about a half hour or so. And then if we have some time at the end, I'll show you guys some of the new features that we have in the e-library. And if there's just anything else you want to talk about, then you're welcome to do that. If you have some questions about research that you're working on, that you want to kind of bounce off of each other, that's great too. So I guess with that, I'll go ahead and turn it over to you, Liana, and I'll turn off my webcam. That way we save enough bandwidth for yours.
All right. Okay, so I'm Liana Nice. I'm a PhD candidate at the University of Minnesota, and I am working on developing a large wild barley integration population. So I'll just give you a little background and then talk a little bit about what my project's about. So some of you may have seen this kind of really crazy schematic of the TCAP grant with all the different populations and things that are going into it. So my population's way up in the corner, and it's the wild barley integration line, so it's one of the, I think they call them like a special population. So yeah, so again, like I said, I'll give you a little bit of background about barley and breeding in Minnesota, wild barley domestication, some challenges to using exotic germplasm, some different populations that have been used to access exotic germplasm in breeding, and then also going into kind of the specifics of my project. Let's see. Okay, so barley, for those of you who aren't as familiar, are working with wheat for whatever. So it's the fourth most produced cereal crop in the world, and there are a couple major categories that are barley is broken into for breeding purposes. So there's 2-row versus 6-row. So you can see here this, let's see. Okay, so that one is a 2-row, and this one's a 6-row where all of the spikelets have filled out and are fertile. Spring versus winter is a pretty obvious distinction, as well as malting versus feed. So feed tends to be higher protein, and malting has, a lot of specific characteristics that are beneficial for the malting process. And in the U.S., feed is actually the largest usage, malting second, and then food and seed production is much smaller. In Minnesota, barley is primarily grown in the northwestern region of the state, and it's primarily spring 6-row malting barley. This area is pretty conducive to high-quality malting barley. But in the 1990s, there's been the emergence of fungal disease Fusarium head blight, also known as scab, and that has caused a really sharp decrease in the production in Minnesota. So here you can see in the upper left corner some kernels that are afflicted with scab. So it causes, the disease produces toxins in the barley spike that can cause problems when feeding it to animals, as well as issues in malting barley. So there's a very low level that is allowed in a barley crop. And in 1993 is when Fusarium head blight started showing up in the upper Midwest. And you can see this is the proportion of the U.S. barley produced in Minnesota. And it has decreased about an order of magnitude since the early 90s. So it's been really devastating to farmers in the upper Midwest. Here's a map of barley production in 2009, and you can see it. Most of the barley that was produced in Minnesota has moved further west into North Dakota and Montana, and where it's drier and the disease isn't as big of a problem. So a little background about the University of Minnesota barley breeding program. So since barley's been bred in Minnesota since the early 1900s, but in 1959 or 58, John Rasmussen took over the barley breeding program and started implementing the advanced cycle breeding process. So this is basically just a process where you cross elite by elite cultivars and maintain a closed population where you don't bring in exotic germplasm or even just distantly related germplasm. And this has led to the production of many highly successful malting barley cultivars, but it also has led to some amount of vulnerability due to the decrease in genetic diversity of the population. So here is a figure that was put together by Conan et al. 2008, and they looked at the Minnesota barley breeding program and put together this really crazy pedigree. And you can see this goes back before 1950, and you can see it's a very interbred population where relatives are being crossed amongst themselves, the elite cultivars by the elite cultivars, and there aren't very many instances of outside germplasm coming into the breeding program. So to add on to this, since this was published, is a new release from the program called Rasmussen, and that is going to be important as it's one of my parents that I'll discuss later. So one of the main reasons this advanced cycle breeding has been very useful for barley is that malting quality is a very complex trait. And this is an example of the American Malting Barley Association's requirements for malt quality in a barley release. And you can see there's many, many different traits that go into it. And they all have very small windows that they need to meet. In addition to these physiological traits, there's other things such as being able to break dormancy, a very tight germination schedule, and even if a variety matches all of these requirements, it may still be rejected based on not having favorable flavor characteristics. So it's pretty difficult to, breed for such a complex trait. So a couple more points about the Minnesota barley breeding program. In 1997, Rasmussen and Phillips published a paper that suggested that genetic gain could continue in this narrow elite germplasm without the introgression of unimproved germplasm. And they suggested that selection on existing and de novo variation as well as further epistatic effects that could be coming out in breeding cycles would be sufficient to continue the improvement of barley. But and they claim that the genetic gap between wild germplasm and elite lines is just too great to start bringing in very unimproved exotic germplasm. In 2008, Conan et al. Took the University of Minnesota barley breeding program and wanted to assess the genetic diversity and how it's changed over the breeding cycles. And they found that, in fact, there was about a 50% decrease in genome-wide genetic diversity within the program. Most of that occurred in the first cycle of breeding, but they also found that the reductions weren't uniform across the genome. So they felt that there probably was still some response to selection that could occur on those regions that are still segregating. So, but there's still some issues. So variation in the breeding program may not be sufficient to withstand novel disease or abiotic pressures. And we've seen this with Fusarium head blight. People are kind of, I guess, less so worried about UG99 as a threat, but that's something that there was very little genetic diversity for the resistance of UG99 in the breeding program. There are other diseases that might pop up, and especially relevant to the TCAP grant are things like climatic stressors and reduced nutrient availability. So my project kind of tries to look at this question of, can we tap the genetic resources from wild barley germplasm for cultivar improvement? And so you can imagine this kind of simple schematic where over time the populations have gone through several bottlenecks, both from moving from a wild species into a domesticated species. You lose alleles due to selection and due to drift, as well as continued improvement will cause this process as well. So we're looking and seeing if we can pull in a few of those beneficial alleles that were lost during the domestication process. So a bit about domestication history in barley. You can see there's some. So cultivated barley is Hordeum vulgare subspecies vulgare, and their wild progenitor Hordeum vulgare subspecies spontaneum. And so they're subspecies, they're very easily crossed, and the genetic diversity of the wild progenitor is much greater than of cultivated barley. And we've learned some about the domestication history through both archaeological and genotypic evidence. And you can see up in the pictures some evidence of barley kernels from thousands and thousands of years ago. So barley was domesticated around 10,000 years ago in the Fertile Crescent area. You can see in the blue dots, let's say, blue dots, so Fertile Crescent area. And then there's also thought to have been a second domestication that occurred in the kind of eastern range of wild barley prevalence. Let's see. So some of the key domestication traits that we have to deal with when we're bringing in exotic germplasm include primarily the non-brittle rachis, or the non-shattering type. So you can see this is a non-shattering type, a domesticated type, and it is fully intact, whereas when you have a shattering type, the seeds are easily dispersed. They fall right off of the spike, and this inhibits harvestability of the plant. So it's a really deleterious trait in a modern breeding program. You can see this individual A is a early domesticate, and you can actually see, if you look at the bottom of the rachis here, it's broken off, whereas in the wild barleys B and D, you can see there's a very smooth point at which it easily dropped from the seed, from the spike. C and D also have this broken rachis area, which is indicative of the fact that they are cultivated barleys. You can also see that C and E, which are much more plump seed as well. These are modern cultivars. 6-row spike is another domestication trait. In E, you can see there's the 3 spikelets are all fully developed, as opposed to a 2-row type, which has the lateral spikelets as infertile. Hulless barley is another up in the left corner. Though hulless barley is less common in Western barley varieties, though it does still occur somewhat in Asia. And lastly, reduced dormancy and vernalization requirement, which are something that may not be quite as obvious to the naked eye, but are very important when you're considering bringing exotic germplasm in, because you have to deal with these cold treatments to get your plants to flower. So, of course, there are challenges. There's kind of an overwhelming amount of crop germplasm in seed banks around the world. So what do you choose to start working with? Linkage drag, so bringing deleterious alleles along with those beneficial alleles that you find in the exotic germplasm can be a big problem. Additionally, phenotypes in exotic germplasm are not always necessarily indicative of genetic value, so lack of domestication traits, epistatic effects, background effects can all confound trait analysis and lead to not really being able to see what wild barley is going to be beneficial for, say, improving yield. So molecular marker technology has been a big piece of the puzzle in utilizing exotic germplasm. You can more accurately characterize populations. It will help develop core collections that will capture the genetic diversity of the whole collection in a smaller, more manageable section. Additionally, there have been advances in characterization, selection, and transfer of beneficial alleles, such as advances in QTL mapping, gene discovery, whole genome selection, etc. And another thing that has been used is the analysis of genetic exotic germplasm in cultivated backgrounds, such as the advanced Backcross and Maize nest association mapping population. So the barley multiparent population I'm developing combines ideas from the advanced backcross populations and the Maize nest association mapping population. So as I've discussed, wild barley has multiple negative traits which can confound trait analysis. So to be able to really characterize the utility of the wild barley germplasm, we're going to need to analyze it in a backcross population. And additionally, the multi-parent part is that single cross populations can't efficiently sample variation available in germplasm collections. So the multi-cross population can allow us to explore large amounts of genetic diversity in a single population. So advanced backcross populations, just quick background, are generally BC2 populations that are used for simultaneous QTL mapping and integration of exotic germplasm. And they allow for analysis of exotic germplasm in the context of leek cultivars. So here's an example where a kind of green, tiny tomato, wild tomato fruit crossed to a normal tomato has led to finding an allele from the wild that actually increases the red color of a tomato. Also, a small berry crossed to a normal berry is led to, even larger, more desirable berries. So there are cases where desirable traits have been found that in wild germplasm and have been brought in through this advanced backcross scheme. So in barley, I have one of these populations as part of my my project. So this is OUH 602, which is a wild barley that's been crossed to Harrington, and it's been previously analyzed for a number of agronomic and disease resistance traits, and I'm taking that one step further to look at yield and malting quality characteristics. So that'll be one aspect of the project I'm working on. So the Maize nest association mapping population, I'm sure a lot of you are familiar with the NAM. They took 25 families that have B73 as a common parent and 25 diverse maize lines, each crossed to B73, and then selfed out into rills for a total of about 5,000 lines in the population. And these lines have been selected to represent the diversity of maize. And this population is being used as a genetic resource for quantitative trait analysis. and QTL detection with high power and high resolution in this joint linkage association analysis process. So one more thing of background. So the wild barley diversity collection is what I've been using to select my parents. So there's a population housed at the University of Minnesota with 318 accessions of spontaneum. They're sourced from 19 countries in the Fertile Crescent, Central Asia, and North Africa. And they were selected based on various ecogeographic characters, longitude, latitude, temperature, rainfall, etc. And they have also been evaluated for several diseases, agronomic and morphological traits. And additionally, they have been genotypes with close to over 4,000 markers. So there's a lot of information about this population available for my project. So my objectives are to develop a resource to increase the accessibility of wild barley germplasm to the barley breeding program, identify and isolate beneficial alleles, examine domestication and selection history of cultivated barley, and then also to characterize the genetics and explore mapping techniques in this novel population structure. So a general outline, I've selected a core set of the wild barley diversity collection. I've crossed these into cultivated barley, and I'm just starting the process of genotypic and phenotypic analysis on this population. So Rasmussen, as I mentioned earlier, was selected as a recurrent parent. It's the most recent release from the Minnesota Barley Breeding Program. And I also selected 25 donor lines selected via power marker software. There was an algorithm that identified an optimal set that would enrich or maximize the allele diversity of those 25 individuals. And that was done using about 2,000 markers. So here you can see the total alleles in the 318 wild barley in blue. And then in red is the percent of alleles captured by 25 lines, and then if I were to bump that down to only 20 lines, it decreases a little bit. So I have about 90% of the total allelic diversity of that wild barley diversity collection captured in this set of 25 lines. You can see it captures SNP diversity quite well as dart diversity, but SSRs are much lower, and that's due to the fact that on average there were around 11 alleles per marker, per SSR marker, so it was much more difficult to capture the allelic diversity of the SSRs. A few ways I can kind of show you the diversity of this population. This is a tree diagram of the entire 318 wild barley in that collection, and in red are the lines that were selected as parents. So you can see it pretty well encompasses the diversity of the collection. I can also look at geographic distribution. This is a map of the Middle East, and here are the 25 barleys and where they were sourced from. So you can see there is a predominance of these Western type barleys in the area around Israel, Jordan, Syria, but I do have a few that are more in the eastern range. Disease resistance of the collection. So you can see the darker the color, the more resistant, and there's quite a range of different resistance to different diseases in the population. So I think I have a pretty good selection of individuals as parents, and I'm capturing a large amount of diversity in this population. So the basic crossing scheme is to take the 25 wild barleys and cross them each to Rasmussen as the recurrent parent. And I've selected a total of 30 individuals per family for a total population size of around 750 individuals. And here's a kind of schematic like the NAM schematic. So instead of just selfing out the F1s into rills, I've actually performed 2 backcrosses, and this, you can see this is quite a bit of work to get 750 crosses in barley. So I don't know if any of you are familiar with crossing in wheat or barley. It's kind of pretty time consuming. So I was in the greenhouse for probably a good 2 months straight making all those crosses. And then I've selfed those out into to inbred lines, and I've finally gotten them out into the field this summer. So the selection of 30 lines was based on that HOUH advanced backcross population I mentioned, and I did just a simple simulation where I chose different numbers of randomly chosen individuals, and 30 individuals gets you about 90% of the wild barley parent genome covered in the set of 30 individuals. So that seemed like a pretty good compromise as far as number of individuals in the population versus the coverage of that wild barley genome in the population. So this is just a crossing scheme. So it's been the last couple years of getting all this population together, and you can see we're now in summer field 2011, and I have BC2 S4 plants in the field right now for preliminary analysis. Genotyping, the parents have all been genotyped with the BOPA1 and BOPA2 SNP chips, so these are around 1,500 SNPs on each of those. And then I'm going to actually select from among that 3,000 SNPs for a custom 384 SNP chip that will allow me to distinguish between regions of the genome contributed by the wild barley parent and the cultivated barley parent. So genotyping will commence in the near future. And then for each line, there'll be some genetic characterization as far as regions of the wild parent that have been integressed, locations of recombination breakpoints, etc. And then the population is going to be analyzed for recombination frequency, segregation distortion, some other interesting population parameters that will maybe be indicative of selection for different traits over time. And then there's going to be extensive trait analysis on this population. It's going to be a resource for analyzing many traits. The plants that are in the field right now are planted in hedgerows, just single replicates, so we can bulk up the seed, etc. So we'll start looking at some things like heading date, height, productive tiller number, spike morphology, and these traits are kind of going to let us explore the utility of mapping within the population and kind of get an idea of what all's out there. But in the future, it is going to go into the TCAP in 2013 and will be fully analyzed for all the TCAP traits like nitrogen use efficiency, etc. So ultimately, anticipated outcomes create a robust resource for mapping and gene discovery, develop a relatively simple way to survey large amounts of barley diversity in one population, identify useful genotypes that may be incorporated into the barley breeding program, and ultimately develop a resource that can help mitigate vulnerability due to narrow genetic diversity. So with that, I'll just acknowledge my advisor is Gary Muehlbauer. I have several committee members, Kevin Smith, Brian Stephenson, Peter Morell, Ruth Shaw, Robert Stupar, have all helped me out a lot with my project. Also in the Muehlbauer, Smith, and Stephenson labs, these individuals have particularly been essential to getting this population all developed. And funding, I'm currently on the USDA National Needs Fellowship, and we'll have future funding through TCAP. So, yeah, I have no idea if any of you are still out there, but you're welcome to ask me any questions about my project.
Thanks, Liana. How about if we give her a round of applause? You guys can do that by your status. I think there's an applause, right? So we can thank. And then if you have a question, you can raise your hand or type it in the chat box. Don Lee has a question, Liana. Go ahead, Don.
So, can you hear me?
Yep.
All right. So, Liana, you've worked a lot already in assembling the population. So, from your experience, what kind of selection happens just in that part of the process? Just in the process of doing what you described, you know, making the crosses and then the back crosses. You know, you haven't even gotten them out to the field yet. Are you imposing any selection, do you think?
Yeah, definitely. So I forgot to mention, I did actually impose selection at the BC2F2 generation for 6-row type because it's very difficult to analyze things like yield, and even a lot of disease traits will vary based on spike morphology. So we decided to actually impose selection on 6-row type morphology, and that way we have a more uniform population as far as measuring yield. So that's one thing. So that was an active selection that we did, but there have definitely been selections. So for example, the shattering trait I mentioned, that's something that we probably lost a few lines because they shattered and we, weren't able to work with them as well. Also, those vernalization requirements and probably less on the seed dormancy requirement, but vernalization requirements are something that we can't really deal with if we're planting it in the field. So we vernalized through the first several generations to try not to impose vernalization requirement selection, but then eventually we had to stop vernalizing. So that's another thing that we probably have lost a handful of lines. We've also been getting some kind of funky stuff that has been kind of fun. So occasionally we'll get kind of necrotic mutants that pop up, and also we had in a couple of the lines they were segregating for some kind of albino mutant where the seedling grew up just snow white and eventually died, of course, but it was kind of fun to see those kind of interesting things popping up. So there's definitely been some selection, but we've tried to minimize it as much as possible.
Cool. Other questions for Liana? Looks like Don has another question. Go ahead while Rebecca's typing.
I'm sorry, I like to ask questions. So how about maintaining diversity in flowering time? Are you able to still do that, or is that very prevalent amongst in the barley germplasm when they start to flower? And did you really have to narrow that in order to achieve your crossing objectives?
No, not really, because I did all of my crossing in the greenhouse, and I planted, I basically just had the Rasmussen parent flowering at all times, so I just, repeatedly grew that up. And so I don't think there was probably very much selection based on flowering time, and there is quite a bit of variation in the field. There's some extremely, extremely early flowering plants, and also some quite late flowering plants. So I don't think there's much selection based on that. Whether or not this field season imposes some selection on that will be to be seen. But right now, based on the population developed up to this point, there hasn't been much selection on flowering time. Yeah, so Rebecca asked how many lines I started with to ensure that I had enough crosses remaining after these selections occurred. So, yeah, that was kind of a difficult thing. I kind of did a lot of learning on my own. So it actually ended up being several several batches. So I would grow up, say, 40 or 50 plants, especially when I was trying to get 30 individuals. And then I'd try to get as many as I could. I also staggered the planting time, so I'd only plant, say, 100 plants at a given period, because it is a lot of work and we could only do so many crosses a day. But if you're interested in like specifics of that, of how I kind of figured out how many I could do a day, if you're doing that kind of thing, you can I can talk to you more about that later. But it was difficult, and I did have to do many more crosses than I actually achieved seed for. So, as far as disease pressure in the fields, I don't think that'll affect selection at all, because the disease isn't bad enough that I'm not going to be producing seed from the plants. Incidentally, there has been some powdery mildew in the field, but that's not gonna really cause any problems as far as maintaining seed. I am thinking about scoring it, but I think it has been sprayed in the field, so I don't know. I probably will score for it because it does seem to be segregating, but it's not a particularly important thing to be looking at. But yeah.
Excellent. Any other questions for Rebecca or for Liana? Oh, here comes Dawn again.
Yeah.
Go ahead, Dawn. I don't know if your mic is muted, maybe.
Yes, that was it. I was muted. So, how does your population look compared to Kevin's breeding lines?
So it looks very crazy compared to Kevin's breeding lines. It's very, very different. Things are all different heights, all different. Some are really bushy, some have like 2 tillers, and that's it for every plant. So if you went and walked through my fields, it would look a lot different than going and walking through Kevin's fields. But it doesn't look horrible. It just looks much more variable, I'd say.
Well, you're probably not impartial. You like your plants because you spend so much time generating them.
Yeah, probably.
I was a postdoc at Minnesota when Don Rasmussen was still there. Have you ever met him?
Yeah, he comes to our seminars occasionally.
It doesn't surprise me. But
yeah, I kind of like the irony of crossing all this exotic stuff into the Rasmussen variety.
Yeah.
It's kind of fun.
He referred to populations like yours as unselected trash. So,
yeah.
but in a nice way.
So, yeah. Hopefully we can get something fun out of it, though.
Well, the tools that you have to look at specific genes and the overall genome compared to what we had 20 years ago, it's amazing.
Yeah.
You'll be able to uncover things that we just couldn't do back when I was a postdoc.
Yep, definitely.
So Liana, after you find these alleles that you're looking for, then what? Will the next step be to actually class it in, or will you clone those genes to transform them into lines? Or?
I don't really know. I probably won't be around for that process, but yeah, I mean, depending on what we find, if it's something really valuable, certainly Kevin's very involved in this project, and I'm sure other people would take it on and either clone genes or start a, more, maybe do some more backcrosses. So one of the nice things I think about this population is, we have all these smaller populations that make up the whole, and say we find that a gene is segregating in, say, 5 of the populations, we might limit our analysis to those 5 subpopulations and do more extensive analysis on just those.
Mm-hmm.
But
that's cool. Other questions for Liana? I did some of my master's and PhD work with RFLP mapping, and that Morex line rings a bell. I got like a Stepto-Morex cross. I don't know.
Yeah. Yep. Morex was in that big pedigree.
Yeah.
So it's back there.
Just be back to memories of grinding tissue. All right. Well, I would like to show you guys a little bit about the learning network. I'll give you another chance if you have questions for Liana. And I'm going to go ahead and share my computer screen here.
Okay, I can get off the webcam if you want to hop on.
Oh, that's okay. I don't want. Yeah, I guess I should, huh?
Okay.
Good point. All right, let me put the webcam on. Guess that's not fair to make you be on it. Okay, so I wanted to show you guys our learning network that we have going so far. Made a little bit more progress on it. So here is your speaker. I don't know if you guys had a chance to see this or not. So we have a section here for webinars now. And so what I'm going to do is make announcements here when we have new webinars coming up. And then I also put links then. I click on the archive webinar. And we've got links to the old ones. So later today I'll go ahead and put Liana's up here as well so you guys can watch that, or other folks that couldn't attend. And what we're going to be doing is setting it up to where I can send automatic emails to you guys to announce, either the webinars or the classes. So, coming up next, we've got in July 12th, we have Rebecca is going to talk about her research with vernalization and photoperiod effects. But what I need you guys to do in order to send you automatic emails is to go into the site. You can just go to pathful.unl.edu/community/pbtn. Let me go ahead and type that in here so you can see what that looks like. And now you can send a link out to all of you as well. And if you can click on this icon here with the lock on it, then it should bring up a screen, to where you can register. I'll send you out instructions how to do that. So I'm already logged in, so that's why it's being a little goofy. But I'd like you guys to get an account with the Learning Network, and then that way we can connect you with any emails. And then also makes it easier for you to go to the discussion board. So we still have that posted as well. So, yeah, I'm not logged in. Sorry. Let me do that here real quick. Looks like I'm uncovering some bugs live, so I always love it when that happens. But what I want to do is kind of test it a little bit more this summer before your official class starts this fall. Alright, so that way we can get all these bugs worked out. Yep, it's not working for me. Alright, let me go in another way. This is part of the e-library, and so that's where I'm at right now. You guys won't have to worry about this. But I work with some other projects, so I'm probably making you dizzy here, going through all of this. Okay, I'm back to the training network now. So you can click on the discussion. I'm crossing my fingers it works. Okay, so now we've got the discussion board here. That's just for the learning network. So we've got some posts in here. So eventually we're going to be putting some topics in here and having you guys discuss it asynchronously for the course. So again, if you can get logged in, click on the icon here, the little lock, and follow the form to fill that out in the next couple weeks, that'd be great. And then, let me go back. So I'm starting to fill in some of the stuff here too. It's about, what our site is about, who the people are. We'll have a link here for the class that will go this fall. And so it will have its own book. So we're starting to put some of the topics in that we're going to be discussing. So it's starting to take some shape. We're not quite done yet with hooking this up to Google and Google Apps, but that'll be hopefully in a couple weeks. So we can use Google Docs for sharing materials and images and that kind of thing. All right, so let me go back here. All right, so that was kind of the stuff that I wanted to show you guys a little bit. Any questions that you have? Okay, Don, do you want to talk a little bit more about the education projects you're working on?
Well, my request for examples of segregation that you might be observing in any of the progeny rows that you're working with this summer is based on one of the things that we'll be doing in our education project. We're trying to target young science learners, young being defined as entry-level college type of learners, and as they're learning basics in genetics and biology, they learn it in a context of how plant breeders use it. So our idea is that might be a first exposure to plant breeders, or to students who might be interested in plant breeding, might increase their interest, and they kind of go from being aware of it to perhaps being interested in it. So actually, I'd like to, if possible, feature any parts of your research stories in these lessons, because that's what makes the most difference to my students, at least, in their interest in learning about it. If it feels real because somebody they can relate to is working on it, it's they're more motivated to learn it. So that was my request for single gene trait examples. I think I'm going to post that up on the discussion board you showed us in the Plant Breeder Training Network. And if any of you have examples and maybe a picture that goes with that, that'd be fantastic. And then we can integrate that into a lesson that'll be a part of the e-library.
Excellent. Questions for Don or other ideas for him? Okay. Anything else you guys want to talk about? Any burning challenges that you're having with your research that you want to kind of bounce off of each other, or any other kinds of questions you have?
I guess I'd like to just compliment Liana on how she gave us some insights with her research story, but I really like the way she started. She showed us how it all fit into the grand scheme of TCAP, which is a pretty grand scheme. So I really appreciated how she got us started in her presentation today.
Yeah, maybe that would be a good model to follow. As each of you guys show your research, maybe you can take that slide of hers and you can show us where in that huge scheme your particular work is at too. Alright, not seeing any burning questions from you guys, so our next meeting we have scheduled is July 12th. If you haven't signed up to present your research, you can do that through the Doodle, or you can just send me an email as well, because I see several of you that are attending haven't signed up yet. So you can do that either through the summer or in this fall as well. So I know you guys are busy during the summer months. Alright, well let's go ahead and give Liana one more round of applause, and then we're done for the day. Thanks a lot for coming and sharing your time with us. And be sure to sign up in the Learning Network. I'll send you reminder emails so you can get in there. Have a good 4th of July.